From Microbe TV, this is TWiP, This Week in Parasitism, episode 109, recorded on May 11th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code MICROBE. Hi, everybody. I'm Vincent Racaniello, and joining me today is Daniel Griffin.
Hello, Vincent.
And that's it.
That's it. No one else. Just us. Well, just us for now.
Dixon is traveling. He's not going to be with us. The first TWiP he's missed.
This is true. After, 108 that he has not missed?
He had 108 in a row. It's kind of like, what was it? All the games going with a hit, Joe DiMaggio, 2000-something game, something, and then you lose it. But he'll be back next time. In fact, next week we're recording again, right?
Yes, we're having a special
episode.
Yes,
we have a guest actually today, but he's not here either. But we're going to start because we have to get this going, and he will join us because he's going to give us today's case, right?
He is, and I believe the next time we record, we will be going to his place, so to speak.
We can't tell you anything other than his name is Paul.
That's it. And, not even a last name, right? People might Google.
No, they might. We don't want you to find out anything about him, but you'll learn next week. And well, we can say that he knows parasites, right?
He's very familiar. He's seen a lot of parasite cases.
Good. Well, when he shows up, we'll just slot him right in here.
Okay.
All right, Daniel, what did we have last time? Remind us.
Yes, let me start by reminding everyone who listened before and actually introducing this to any new listeners who are listening for the first time. We had a case of a 26-year-old male who was a long-haul truck driver in India. He was driving all around. He's actually from the northern tiger region of India, the area where there's tigers. They're very proud of that. And he had come into the hospital at the end of the rainy season with 6 days of fever, chills, muscle aches, small loose stools, vomiting, trouble breathing, cough, and decreased urine output. He had let us know that he lives in a mutt hut with a coconut leaf roof. We had actually contrasted this to how many people were excited that they were living in concrete homes. So this man, in the economic spectrum is down lower on the end there. No one else in the family is sick. He does live with his wife and 2 children. They're currently well. He has significant animal and insect exposure. We pointed out that there were dogs, cows, monkeys, and as a long-haul truck driver, he's out there and just sleeping, no mosquito netting, etc., and sleeping all around India, so he's getting exposed to a lot of different locales. He had gotten sick after coming home from a trip up north. He said prior to this he had no significant out-of-the-ordinary medical problems. He'd had no surgery, and now he is really sick. He says he has pain everywhere. We got a little bit into his social history and found out that he occasionally drinks palm wine, rather popular in this area. We do notice that he's got some yellowing of the eyes, and we weigh him. His weight's down a little bit, and this is actually decreased from previous weights. He has this fruity breath, which we described as ketonic. On exam, he has a fever up to 39°C. It's well over 100 Fahrenheit. Blood pressure is 100/70. His heart rate is increased in the 120s. He's breathing rather rapidly in the 20s. He looks like he's in a lot of trouble. He looks distressed, and cognitively he's not really that sharp, a little decreased as far as his level of alertness, his mentation. We don't really notice a lot focal on exam except his abdomen. His belly is tender, especially in the upper right region, and he has a enlarged, palpable spleen, so I can actually feel his enlarged spleen. As I mentioned, the upper right, I can feel that there is tenderness when I push where the liver is, though the liver does not seem to be enlarged. And we had some labs that we gave out. His BUN, blood urea nitrogen, was elevated at 102. Normal's up to about 20, so that's quite elevated. His creatinine, which tells us a bit about kidney function, is also elevated. His hemoglobin is a little bit decreased. Say normal's about 15, he's at 11. His platelets, normal platelets should be about 200,000. He's down at about 9,000, so quite low. His white count is 10.3. So just sort of upper limit of normal, maybe a touch over. But we noticed he had 0 eosinophils. So as they like to say, eosinopenia. His LDH is up at 8,000. That's very high, about 40-fold higher than it should be. AST, ALT, those are our liver function tests, or I should say liver inflammation, but we call them liver function tests. Those were normal. Bilirubin was significantly elevated at 21. And as he tells us, he has never been this sick before in his life. I'm always entertained by that. I've never been this sick before. And you know what? In this case, this guy's right. He's never been this sick before.
All right, we have some guesses. First one is from Mark. Dear TWiP Trio, you have me very stumped with this case. Patient lacks eosinophils, which makes parasite more likely a protozoan. Renal failure, hepatic tenderness, normal liver transaminases, but increased LDH, profound hypoprothrombemia, anemia can be symptoms of malaria, as splenomegaly. Patient's high lactic dehydrogenase may be a result of hemolytic anemia. WBC is low or normal in malaria. Regarding ketotic smell of breath, patient probably has insulin-dependent diabetes mellitus. Now, malaria can cause hypoglycemia and also in rare instances, pancreatitis. And pancreatitis may cause diabetes or a decreased production of insulin, so that body relies on alternative pathways for energy that led to ketosis. This is a far stretch. More likely, ketosis could be related to binge drinking of palm wine and other alcoholic beverages, which could cause alcoholic ketoacidosis. Don't believe ketosis is related to parasites. I have more questions than answers, and I'm probably wrong, but I am guessing Plasmodium falciparum for this interesting case. I look forward to the answer for what is afflicting the young Indian gentleman and his outcome after diagnosis and treatment. If the patient has malaria, treatment options depend on susceptibility. India is kind of in a gray zone as far as chloroquine susceptibility. Patient may have a serious case of malaria if he has malaria because he has renal failure. Recommendation per CDC is IV quinidine gluconate plus clindamycin or doxycycline or tetracycline. Gives a link for that. Don't know how available these medications are in India, but I have seen Dr. Reddy products in my pharmacy. Dr. Reddy is a large international drug manufacturer based in India. Temperature today in Oklahoma, 85 F. It is spring, but feels like early summer. Global warming seems real where I live. Mark. All right, take the next one.
Yeah, I only wish global warming were regional, right? Wouldn't that be nice?
Yeah,
so Ruben writes, Dear TWiP experts, as I heard the case, I was immediately struck with the patient's severe eosinopenia, since I knew that visceral leishmaniasis may be accompanied by eosinopenia. However, I suspected that Daniel is unlikely to present the second visceral leishmaniasis case in a row. Another person, say from another podcast, could have done something like that, but not Daniel, who comes across as a really decent and caring person. I feel boxed in now. You know, I can't change the diagnosis. This isn't like battleships where. So I'm hoping at this point it's not leishmaniasis. Now, anyway, that thought left me with a loud and clear diagnosis of severe malaria with the cerebral syndrome caused by Plasmodium falciparum. There is a small chance that it is caused by P. vivax, but it is far less typical, and the treatment is going to be the same. I learned a bit about malaria from Victor and Ruth Nussensweg during my tenure as an associate professor at NYU and Courant Institute of Mathematics in the 90s. Severe forms of malaria can develop very quickly and may lead to death. The clinical features of severe malaria are listed below match all the symptoms and test results described by Daniel and include jaundice, elevated bilirubin, kidney dysfunction, anemia, fever, and impaired consciousness. The latter may transition into a coma. The most intriguing feature, eosinopenia, is also found in severe malaria. According to some authors, the eosinopenia is not due to underproduction in the bone marrow, but rather is the result of acute inflammatory response. Furthermore, eosinopenia may be associated with severity of malaria, and it was found that eosinophilia associated with an Ascaris co-infection is protective against cerebral malaria. Treating this patient is tough as he may be about to slip into a coma. The patient needs to be treated with the IV infusion of antimalarials and possibly anticonvulsants and antipyretics. They may be treated with parenteral artemisinin derivatives such as artesunate, artemether, alpha, beta, artether, an ethyl ethyl derivative artemisinin for at least 3 days to see progress. Quinine is another option, but it may be dangerous. I hope that with Daniel's help, the patient dodged the fatal outcome. As a disclaimer, I have to say that everything written above may be utter nonsense since I am a physicist, mathematician, and never studied biology, medicine, or parasitology properly. Keep up your good work. It is a lot of fun for all of us. Ruben. And Ruben Abagyan is a PhD. He's a professor at the University of California in San Diego at the Skaggs School of Pharmacy and Pharmaceutical Sciences, San Diego.
2 malarias so far.
So far, yes.
Brett writes, Hi TWiP, after listening to you guys for the past 3 years, I figured it was finally time I write in. I'm in the last month of my getting my bachelor's in cellular and molecular biology, and I'm frantically trying to finish a project in my lab. Which brings me to why I started listening to your lovely podcast. It's an avian parasitology lab at San Francisco State University. We're focused on avian malaria, but my project looks at diversity and prevalence in the islands off of Papua New Guinea. I had no prior knowledge of parasites when I joined the lab, and TWiP has helped me immensely over the years. Thank you. So let's see if I'm close at all with my diagnosis. My guess, this could be a double infection or triple. Of course, my initial thoughts go to malaria. I admit I am biased for that, but I don't feel like that would explain the dry cough and decreased urination. But aha, this is where I think I land my breakthrough. Could this be a schistosome infection, perhaps with malaria? And finally, the last option visceral leishmaniasis. My logic is as follows. Schistosoma japonicum, although normally found in China and in the Philippines, I think could have come into the northern part of India and possibly contaminated the water he drank, especially if just after the rainy season, fecal contaminants or possibly even the snails themselves were flushed into a drinking source. I couldn't find any literature on whether the Oncomelania, the snail intermediate, is in India, but it is in China. Plasmodium falciparum. Fever, chills, vomiting, and general body ache in India will always make me think of malaria. The acute and virulent nature of this make me think it's falciparum. The thing bugging me, though, all of this case was the eosinopenia. Wouldn't either schistosome or plasmodium infection lead to an increase in eosinophils? I asked the professor in charge of my lab, Dr. Ravinder Segal, and gave him the symptoms I could remember offhand. He agrees with my P. falciparum diagnosis, but thinks schistosomes would only be caught if he was swimming in the contaminated water. But another aha moment, he suggested that it could be visceral leishmaniasis, as this would explain the swelling of the spleen and liver, as well as the weird blood cell counts. To confirm one of these, I would run an ELISA for both leishmania and schistosoma japonicum, as well as do a blood smear and GIMSA to see if one could find plasmodium. This could also be done using PCR, but a blood smear can be done quickly and cheaply. You all have been a great inspiration to me over the years, and although I'll be moving to Ann Arbor, Michigan for a master's in molecular, cellular, and developmental biology, I'll still be in contact with my lab to discuss the latest TWiP. Keep up the wonderful work, and sorry for the horribly long email. Brett from San Francisco State. And that's it. 3 guesses.
Yeah, we a lot of our usual...
Not here.
Not here? I don't know. Are they on vacation? Is there a vacation?
I don't know. Maybe the end of the school year is trying, people are busy. I know I've been busy.
I've been busy.
So that's all right. So they all have malaria, and the last fellow has a multi-infection, huh?
Yeah, well, let's go through. One thing I noticed is as we read these, usually Dixon is moaning to my left every time he hears something that he doesn't agree with.
Mm-hmm.
And fortunately, usually the mic is away from his mouth, so we don't all.
He does have color. I'm sure he would have had comments on.
He will have. So let's go through a few of the comments, and then I'll do as we say, the unveil. So we'll start off with the first gentleman here, and he is he's talking about the lack of eosinophilia, right? And that draws him to think about a protozoan infection. And I think people are listening, people are learning, because that's whenever I get a white blood cell count, I always ask what's going on with the eosinophils. Are they up? Are they normal? Are they down? When they tend to be up, that makes us think about our tissue invasive helminths, our worms that are invading. When we have a complete lack of eosinophils, then we think about these severe protozoal, like we'll say a falciparum, a malarial type infection. Or, in all honesty, in this country, whenever you see a lack of eosinophils, it gives you the heads up that they're really sick. Usually these are being cleared because of just such a robust immune response. So in this case, we're thinking less about our worms, more about our protozoal infections, and we're thinking that this guy is right. He is really sick.
He's got more symptoms out than you would expect for malarias, right?
So this is actually, and I'll cut to the case a little bit. So I saw this guy with David Worrell, and some of our listeners probably know David Worrell. David Worrell is out of Oxford, but and I think the out of Oxford is the thing I talked to him. That's where his professorship is, but I don't know how much time he's spent there because he's always off, throughout the world in these tropical sites and seeing these cases. And so I see this case with David Worrell, and he's focused on this man's severe abdominal pain. Right, so he's got a fever, but what he's really complaining about is his belly doesn't feel well. He's got loose stools. And so, he's giving me what causes abdominal pain and fever in the tropics? And I came up with my idea, and he gave me a hard time about it. But we'll get into that. So a few more things that people bring up that help steer us in on where it is we have a large spleen. The liver's tender, but the liver's not enlarged, and I think that's critical too.
That there's clearly some liver damage because the enzymes tell us that, right?
Exactly. So there's inflammation going on, but the liver's not enlarged. And so who was it that wanted me not to present another case of leishmaniasis? Your visceral leishmaniasis is going to give you an enlarged liver.
All right, so we can cut that out immediately.
We could probably decrease. And also, I'm a nice guy, so we have to leave that out right away because
I don't know why being caring and decent would prevent you from having 2 cases of leishmaniasis in a row.
I don't know.
It's not you get what comes to you, right?
Yeah, exactly. And the other thing I will say, so that leaves us with, here's a gentleman, he's in the tropics, he's really ill, he's got a high fever, he's got a large spleen. That's malaria until proven otherwise. So even though he's got this, his abdomen hurts. And that was what David Worrell was giving me a hard time about. He's like, but he's got severe belly pain. Why don't we scan his belly? What's wrong with his belly? And I think that's the thing in the tropics. We say until you have a negative blood smear, they have malaria till proven otherwise. When they have fever and a large spleen, they have malaria. And so that's sort of
very good.
And so in this gentleman, we actually went ahead and we did a blood smear.
Okay.
And the next thing, so it's going to show malaria. 25% of his red cells, a quarter of every red cell, is parasitized and multiple forms.
Wow.
And so this man actually had a mixed infection with falciparum and vivax.
Wow.
Now, the interesting thing, and this brings up something interesting if you're in the tropics. If you go to a place in Africa, because it's very endemic there, a lot of parts of Southeast Asia, we'll say India, and you do blood smears on everybody.
Right.
In the town where I was at this hospital, a certain percent of the people, a large percent of the people, if you really look closely, you're going to see malaria forms in the blood. And this is that low level of malarial infection that maybe gives you some sort of an immunity, a resistance. But what got this guy in trouble is he was living in an area where Vivax was endemic. He probably had Vivax all the time. Now he gets infected with falciparum.
And his travels, right?
And his travels. And the falciparum is not something that you tolerate. And so that was what got this gentleman incredibly ill. And
but what's the belly pain from?
The belly pain is probably related to a few things. One is probably an acidosis that's going on. There may be even an ischemia. Probably he's not perfusing his belly particularly well.
Is breath also part of that?
So the breath was coming, and this is tied in from a hypoglycemia. He's running out of sugar, so he's switching over to ketone metabolism.
I see. Because the parasites are using it all up?
That's actually what we think. You think basically they got to eat too, right?
Yeah,
and they're eating all your glucose.
Wow.
And in some cases, depending upon which treatments, the treatments can worsen the hypoglycemia. So you really have to be careful with these people. In addition to, as we talked before, about needing a lab to make the diagnosis, you need a lab because you're going to be repleting the glucose in this individual and giving fluids because of the kidney failure.
Wow.
So this gentleman got IV artesunate, which brings me to the gentleman who looked at the CDC website. And this is a bit of a sticking point, I think, for a lot of us who care about people that get malaria.
That was Mark, right?
Is that everywhere in the world, except the U.S., pretty much, uses IV artesunate. And there's a number of head-to-head trials showing that at the IV artesunate preparations, you get faster parasite clearance. You actually have mortality benefits. But it hasn't made its way through the FDA in the U.S.
Interesting. They want to use quinidine gluconate.
So that's what they make us use here in the U.S.
Does it work?
It works. It works.
Would it work in India?
So it would probably work. It actually works everywhere.
Everywhere, okay.
Yeah. So you worry about the quinine resistance. People sort of threw that in, but you really don't. You're using high doses. It's going to treat. It's just not going to treat as well or as quickly as the IV artesunate.
Why did they give antibiotics as well?
So we say there's two, and you and I were at a talk, Vincent. Eventually, we're going to say there's three. So you want one really quick acting agent, and that's going to be your quinidine or your artesunate. But then you want a longer acting because otherwise resistance is going to develop. That short acting agent is going to wear off, and then you're going to come back with resistance to it. So you add a second drug. It could be clindamycin. It could be doxycycline. In this case, it was doxycycline. And I think as we heard about, maybe you want to add a third drug because we're having a problem.
Yeah. Right.
So
amazing. Do you know this Dr. Reddy, by the way?
I don't actually.
You haven't heard of it, huh?
No.
I'm surprised since you go there often.
It's
right?
probably, what is it? Probably written on the medicine.
I'm sure.
I'm Googling right now.
So everyone got falciparum, but no one got Vivax. Although one person thought it was I
think it was one person
co-infection with schistosome, right?
Yeah, it's interesting. We have this mantra, I guess I'll say, that all really sick fatal malaria is due to falciparum malaria. And I'm gonna say that that's not true. If you look at the cases that we see here in the U.S., the majority are falciparum, but 10 to 20% are not, and you can get severe malaria from Vivax. So the person who brought up Vivax, that's reasonable. Some cases of Vivax, these are going to tend to be in your non-immune, someone traveling to an endemic area who hasn't built up this immunity.
Yeah, that's right. Ruben suggested Vivax, but didn't go with that.
There's a small chance.
Yeah.
And I think he's right. I think the illness is caused by Plasmodium falciparum. There's some Vivax mixed in. I don't think that's what's causing the problems here.
So what you said was fever, enlarged spleen. In India, they have malaria.
I'd say in the tropics. If you're in a malaria endemic region and they've got a large spleen, and actually the WHO uses that as criteria. If you don't have any lab, you treat them for malaria with a fever and a large spleen.
Reminds me of the first case definition of AIDS, which was Kaposi sarcoma. I don't know, opportunistic infections. I think it was just two things.
Yeah, it didn't. Well, I mean, if you look at Kaposi sarcoma in particular, it's very uncommon to have that. Outside of that, there's that small demographic, right? But otherwise, it's if you see it on the face or not on an area other than the lower extremities, other than the legs. Yeah, even in today's world, it's almost always going to be HIV.
Yeah. How did you treated him with IV artemisinin, right?
He got the IV artesunate, and then the doxycycline was added. Added actually pretty quickly. David had a comment about that. It's in this area. You also have a lot of scrub typhus.
Yeah.
So you don't need to wait to add the doxycycline, right? So you add it. It gets added on a little quickly, and maybe that also covers in case they've got something else going on, which in this case I don't think he had. And the other thing to worry about is sometimes these people will actually have a bacterial sepsis in addition. This gentleman, fortunately, just the IV artesunate, the doxycycline very quickly got better. And I think at 24 hours, his parasitemia was down to 1%.
Very good. He must have felt better, too.
He was a lot better, yeah. He was a lot better when we went back.
Now, Ruben said that quinine, it may be dangerous, but of course that's what the CDC recommends. Now, why
Quinine's dangerous for a couple of reasons. One is, as I mentioned, the hypoglycemia. It is notorious for causing a problem there. But also, whenever we give the IV quinidine, we always put the people in the intensive care unit because of the arrhythmia problems, so the heart rhythm problems.
All right.
So yeah, it was a good case. And it was good because he got better.
Dixon didn't tell us what his guess was, did he?
Dixon didn't tell us.
Of course, and I would text him, but he doesn't really read his text, And
I think the areas where Dixon would have maybe cringed, and I think it's probably important to bring this up, is we had Schistosoma japonicum brought up. And the person brought up that he could have possibly contaminated water that he drank.
Right.
But I think we have to remind people that you don't get schistosomiasis from drinking contaminated water. You're actually going to be, I think, as his professor in charge of the lab said, you end up swimming.
Right.
So you're in the water. The water's on you. These little metacercal forms. They're actually going to penetrate they're coming in through your skin.
Right.
And what triggers them actually come out of the water. And as you start to dry off, just as these little droplets are starting to dry, it triggers entry. So when I was swimming in Lake Malawi, we toweled off vigorously.
Wow. All right. That's great. Anything else on that before we move on?
No, I think
that's it. Good.
I think that was it.
Now, if Paul doesn't show up, do you have a case that you can use in case?
I could grab one, but I actually just got a text. Paul is on his way.
All right.
He is he's down, I think, by Grand Central.
Okay.
He took the shuttle across, and
he probably will get here when we finish the paper.
Actually, that's perfect, right? Then he could throw his case.
That's fine. All right, the paper is in Science, which Daniel picked. It's a very cool paper. Parasites resistant to the antimalarial atovaquone. Atovaquone failed to transmit by mosquitoes. I won't ask you to prevent. Atovaquone.
That's what we call it.
Failed to transmit by mosquitoes. Number of authors. The first is Christopher Goodman. The last is Jeffrey McFadden. And they are from Australia, Baltimore, Bloomberg School of Public Health, Indonesia, and Tokyo. Wow, that's quite an international collection of authors there.
It's great to see that. Isn't that what we always talk about? We want cooperation and collaboration in research, and we've got it here.
We always talk about how in various microbes and parasites, if using a drug and getting resistance has a fitness cost on the organism, which would be good then, because then it can't spread as well through the human population. And this is a wonderful example of a clear fitness cost, at least in mice.
Yeah.
Now, this drug, atovaquone, we haven't talked about this at all, have we? It never comes up in any of our cases. Did you ever use it?
So I've not only I've taken it myself quite a bit.
It's taken as a mixture, right?
It is. It's taken, people may know of it as Malarone. Okay. It's the combination of atovaquone and proguanil.
Okay. So when you get ready to go, you take it here ahead of time, or?
So the great thing about this drug is you can start it a day before you arrive somewhere, and you only take it for a week when you get back. And it's a great segue into the different, say, preventive medicines for malaria. A lot of the preventative medicines for malaria actually don't prevent you from getting malaria. They prevent you from getting sick. So they only work on, we call it the erythrocytic or the red cell state. So you still get infected. The malaria parasite still goes through the liver. It ends up in the red cells. And then what you do is you're treating it. So when you come back from one of these areas, if you've been infected, you may actually be treating, not protecting yourself, but treating yourself for 4 more weeks so that it gets eradicated. This is the problem, though, right? You come back, you're back from Africa, you feel fine, you've been taking this stuff for a week or so, you stop. and then you get sick. Now, the nice thing about this Malarone, the atovaquone proguanil, is it actually affects the parasite probably in the liver phase as well. Mm-hmm. So it probably really blocks things up front. It probably prevents you from getting much of an erythrocytic state. So you only need to take it for a week is the recommendation, but some studies say maybe 3 or 4 days after you get back, and then you're done.
Yeah.
But you know what? It costs a fortune.
Yeah,
You got to take it every single day.
Yeah.
So when I was in Malawi last time with family, my sister-in-law, brother-in-law, and their kids, every morning everybody would line up their Malarone, and you've got the kids' dose and the adults' dose. We'd all take our Malarone with breakfast. It was very entertaining.
It's interesting you say it's expensive because they say now cheaper generics are rolling out. So there's going to be more extensive use, and you do get resistance, and they figure, well, we're going to get more resistance because cheap drug equals more resistance, right?
Exactly.
Inappropriate use. So they wanted to know if there was any fitness cost associated with this resistance. And the reason they ask this is because the target of atovaquone is cytochrome B, which is encoded on mitochondrial DNA. So it's a little bit unusual. And as you'll see, the selection is different in different stages of the life cycle. So they do a simple experiment. There are known alleles for resistance. And they ask, can malaria with these mutations transmit from mouse to mosquito and back to the mouse? Mm-hmm. So they use a mouse model, Plasmodium burgii, and they use Anopheles stevensii mosquitoes, both of which we've mentioned, right, on the show. They have 3 different mutations that are known to confer resistance to this drug. So basically, they infect mice, and then they allow mosquitoes to feed on the mice.
It's really terrible, right?
Well, that's what lab mice are for, right? And then they see if the mosquitoes can transmit to new mice. Mm-hmm. So all 3, so they have 3 parasite lines, each with a different resistance mutation. They all produce wild-type numbers of male gametes. Parasites with 2 of the mutations, then again, the mutations are in the cytochrome B gene. They can self-fertilize, make ookinetes, and produce oocysts, but the oocysts they have developmental defects. Mm-hmm. So they can't go on from that point. And parasites with the other mutation were defective in the ability to self-fertilize and infect the mosquito host. So in other words, you have these mutations in the mouse, they can't infect the mosquito. They did 17 attempted mosquito infections for mice, right? No parasite carrying an atovaquone-resistant cytochrome B mutation was able to generate sporozoites in the mosquito salivary glands or was able to infect the naive mouse. So they say rodent malaria, resistant mutants are unable to transmit from mouse to mouse via mosquito. So these are 3 mutations that they say they're clinically relevant mutations. They find these in people.
Yeah,
right?
Well, yes.
But of course not in burgii, right? It would be
in falciparum.
Falciparum,
yeah.
Right? But I guess the cytochrome Bs are similar enough that they can say here.
Yeah, actually very similar. And I think as you're doing this, I'm realizing, to jump in and cover a little bit of Dixon's spot here, right? Because he's going to make sure we all know the life cycle.
That's right.
So we've got an infected person, and they develop the gametocytes, basically.
Right.
And then the mosquito is going to take those up, and they're actually going to mate. The male and female are going to mate in the mosquito, and it's going to produce this progeny, which is going to ultimately end up with the sporozoites that they can infect. And what we're seeing here is that in us, in mammals, you can go ahead and select for these mutations, and there doesn't appear to be a problem because of the different metabolism requirements. But once they get into the mosquito and they need the full-blown aerobic respiratory cycle, it fails. And you end up with these deformed, ineffective eggs, basically.
So the
oocysts.
Oocysts. Those are the eggs that would then go on to develop
into the sporozoites, which would then
be transmitted from the mosquito salivary gland. Yeah, it's very interesting. They're just totally defective and can't be transmitted. So then they ask, well, if you had a a sensitive parasite infecting the same animal, would they somehow complement, right? And
they're
recombine?
trying to do these crosses to see if somehow they can.
So can they if you crossed a resistant and a sensitive parasite, could that transmit the gene? So they made crosses of resistant and sensitive parasites, and they note that this would simulate what happens if a mosquito would bite someone that's infected with both resistant and sensitive parasites, or two different people. They would mate, right?
Yeah, I mean, that's the interesting thing, right? We think about mosquitoes, and they're going to bite a person, but most mosquitoes, 99% of mosquitoes, only take one blood meal once, right?
That's right?
So this is sort of this double biting effect. They're actually this,
Yeah, we talked about it on TWiP because they take a blood meal, they're kind of heavy, and they go rest somewhere for a while.
And they like that. They like to rest on
surfaces. They don't want to immediately bite someone else, right?
Yeah.
Someone said typically on a windowsill, on your windowsill, they'll be resting after they bite you.
Yeah.
So if you had a wild type and a resistant parasite in the host, for example, could they recombine? Could they mate and make resistant? So maybe the defect imposed by resistance could be somewhat complemented by having the sensitive. Yeah. So they they make a cross, and then they put the crossed parasites into their assay, feed them to mice, and they show that the mice can get infected, and they can show they get outcrossing in the mice. In other words, the resistant and sensitive parasites are crossing. They can do that by looking at the alleles, but all 14 mice carried parasites with only wild-type sensitive alleles. Outcrossing did not facilitate transmission of resistance. So in other words, no outcrossed parasite was transmitted, only the ones that were still sensitive. So the concern that having double infections will rescue this transmission defect isn't an issue at all. They also did cool experiments where they label the sensitive line of parasites, sensitive to the drug, with a green fluorescent protein, so they can watch it in these crosses. And they cross that with the 3 resistant lines, and then they can track sensitive and resistant parasites in that way. All the progeny had wild-type cytochrome B genotype and were sensitive to atovaquone, reaffirming that resistance cannot be complemented and transmitted even when one parent carries a wild-type version of the gene. So that's the key, right? Having either 2 mutant or 1 wild-type and 1 mutant gene, and mutant meaning resistance, you can't transmit. Yeah. If only it were the same for all resistance in all cases, for not just malaria, but bacteria as well, right?
Yeah.
Wouldn't that be good?
It's nice that there's an upside to the fact that these parasites have become resistant.
Well, in this case, it's really good. I mean, but I think you would know that. I mean, is this resistance not spreading globally? Is that I mean, you would predict that from this work, right?
Yeah, I mean, from this work, you would say that the only people that are going to get resistant are people who they in themselves have been treated. It's not that you're going to, it's nice here. You're not going to catch atovaquone resistance. And,
I guess it can still be an issue, right? Because you can treat people and they develop resistance. They just can't transmit it. So it can't spread as rapidly, I would suppose, which is good.
Yeah, but I mean, it's still a problem if you've got a patient, you're trying to treat them with this. Fortunately, we do sometimes actually treat with this, and it'll be sometimes, the Swiss are big into this, where someone's traveling to an area, maybe they're going to be there for a while, and they're not excited about, as I mentioned, the morning ritual, taking their medicine every single day. So they basically have a emergency treatment pack and one of these little instant malaria tests. And so what they do is they don't feel well, they test themselves, and if they're positive, they self-treat, and they self-treat maybe with Malarone. People tend not to make good decisions when they're febrile and sick with malaria, so there might be a downside to self-treatment, but you could end up potentially with a problem.
The next experiment I think is very cool, and this is based on the fact that the cytochrome B gene is encoded on mitochondrial DNA, which is maternally inherited. You get it from females. And so, in other words, females are supposedly transmitting resistance because it's on mitochondrial DNA. So this would predict that the mutation would render females sterile.
No, so what I like about this is there's always this big thing is that no matter what bad happens to you, we always blame our mom. Well, in this case, it's true.
Blame your mother.
So, these basically the inability of these to survive in the mosquito is coming from the
right
female side.
So the fact that these gametes can't survive would suggest that it makes the females sterile.
Yeah,
because they're the ones that's transmitting it. So they do an experiment to prove that. They cross their 3 atovaclone-resistant lines, which with parasites that have been modified to be either male or female sterile.
Mm-hmm.
And the idea is if resistance is linked to mitochondrial inheritance, they would get normal recombination to male-deficient parasites, but no progeny with females.
Yeah.
And in fact, that's what they see. Crosses to male-deficient line, they got recombinant progeny, although they can't spread because, again, the phenotype of this mutation is that it can't spread from mouse to mouse by mosquito.
Yep.
And the female infertile line, no progeny, 16 out of 17 attempts to transmit to a mouse. So these resistant mutants are female sterile. The females cannot reproduce, which is very interesting. 44 separate transmission attempts involving 750 mosquito bites. Transmission of resistance was only observed once, and this mutant didn't transmit further. So once out of 750, that's not bad.
No, that's not bad at all.
All right. What else? Oh, so now this is all done in mice, right?
Yes.
What about people? So they select a P. falciparum atovaclone resistant lines by passing them in the presence of the drug, and they got 2 mutations in cytochrome B, and they fed them to mosquitoes, and they don't have mice here because I guess mice, you can't do P. falciparum in mice.
No, and that's the interesting thing is the different strains of malaria are sort of host specific.
But what they can say is that the resistant mutants are impaired in mosquito infectivity and the number of oocysts produced. So they say probably wouldn't be able to infect people. But I guess, epidemiologically, you could look at patterns of transmission of resistance and make that...
because you can look at people who are newly infected and, say, are they actually infected, or are we only seeing, the time course? Are we only seeing the resistance after treatment?
So do you understand the logic of why mutations affect the mosquito stages and not the mouse?
Yeah, I mentioned that a little bit earlier.
Yeah.
There's different types of metabolism. And so when I wanted to say I'm anthropomorphizing the malaria, but during the stage when the malaria parasites are in the mosquitoes, they require this full aerobic respiration,
right?
And also this sort of active TCA cycle. So they're really metabolizing, and that full metabolism is required to go from an oocyst to these infective forms. Once they're in a human being, it's not required at the same level. They're actually operating at a lower metabolic level, even though, as we pointed out, they're sucking away all our glucose. And so when you put them in the mosquito, it can't get to that critical level, and you can't have the proper development.
So in the mosquito, you need cytochrome B.
And you need it at a full level for the full aerobic metabolism.
And that's why these mutations, they compromise its function, whereas in mice, it's not as critical. Yeah,
they're not 100% aerobic glycolysis dependent for their...
Right. Transport is essential, but in mice it's easily overcome. Let me see, where do they say that? I want to find this.
I think when you get near the end, they talk about how the ubiquinone cytochrome B electron transport operates at only minimal level, while the malaria is in the blood phase.
That's right. That's right. In the mosquito, they're in active, full aerobic respiration, as you say, with an active TCA cycle, robust electron transport. And then, in that case, the respiration deficits of the mutants prevent them from completing their generation of sporozoites. So cytochrome isn't a unique target in that sense.
It really is interesting because there's different requirement whether it's in the mouse or whether it's in the mosquito.
Our guest has just arrived, and he's joining the conversation. All we're going to tell you is that his name is Paul. Welcome to TWiP, Paul.
Thank you very much for having me. I'm sorry I'm late.
No problem. And Paul knows about parasites. That's why you're on TWiP, right?
Exactly.
Knows and loves parasites, right?
Next week, we'll tell you everything about Paul, but today we can't because it would give away the neat case that he has. We were just discussing this very cool paper in Science where they showed that parasites that are resistant to atovaquone, is that how you say it? Atovaquone?
No, not that latter one. You were doing well.
Atovaquone can't transmit from mouse to mouse by mosquitoes because they have a defect in cytochrome B, and that makes them female sterile.
And the most amazing thing about that, it's the only example I know of about a drug resistance in anything that self-eliminates itself.
That's right.
When you look at the problems with bacteria, viruses, fungi, parasite, chemotherapeutic resistance, they're all a problem because of their resistance, and this solves that.
It's amazing.
Which is it's the only case I know of. It's quite interesting.
So we were just saying, can we find similar targets like cytochrome B for other inhibitors?
Yeah, and
right?
these guys actually suggest that because I think the novel thing here is that parasites live in two very different environments, sometimes more than just two, right? We see with our flukes, for instance, they might be in a snail, they might be in a fish, they might be in a person. And because they live in different environments, what they might do that has, we talk about a minimal fitness cost, as we're seeing here, maybe this has a minimal fitness cost to make a cytochrome B mutation to escape the drug in a human, in a mammal, and a mouse. Yeah. When they then get into the mosquito, that's an absolute fitness cost. They can't go forward at all. So what they get selected for in one environment, when they go into the other environment, be completely lethal.
That's the key. Different selection in the mosquito versus the mouse.
So parasites may be really unique in that we could knock them all off in a few weeks. Now that we know this.
Well, they say at the end here that people are looking for other drugs that target cytochrome B, and that makes sense, right?
Yeah, and then they even mention going after maybe certain components of the apicoplast,
which are maternally inherited. That's right.
Yeah.
The cool thing about mitochondrial DNA genes is there's no recombination.
Yes.
So you're not going to get recombination with sensitive alleles. That could be a problem. So that's pretty cool. It's a very neat paper. Did he tell you we were doing this beforehand? Yeah, this is cool.
Yeah, it was very interesting.
Anything else, Daniel?
No. I think that we'll hopefully see a lot more, capitalizing on this concept and helping fight these parasites.
All right. Now, Paul, you came just in time.
Perfect.
Because do a case study. But before I do that, I have to do an ad. This episode of TWiP is sponsored by CuriosityStream, the world's first ad-free nonfiction streaming service, founded by John Hendricks, the founder of Discovery Communications. They have over 1,400 titles and 600 hours of content. You can get it in 196 countries, and it's available on many platforms, including on the web, Roku, Android, iOS, Chromecast, Amazon Fire, Amazon Kindle, and Apple TV. They have a wide variety of science, technology content, also nature, history, and many more topics. A lot of 4K content, which they've just launched. One of the largest 4K libraries on the internet. They also have interviews and lectures, things like Stephen Hawking's Universe, The Next World featuring Michio Kaku, and they have viruses as well. There's a new video on Zika virus and many others as well. Basically, with CuriosityStream, you get real science shows, not just reality TV science shows like the ones plaguing cable TV. There are monthly and annual plans available, and they start at just $2.99 a month, less than a cup of coffee, or the cost of one title on competing on-demand platforms. So check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 months free of one of the largest 4K libraries around. Go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their support of TWiP. So we have this tradition ever since Daniel has joined us. We went for a while on this podcast, and then Daniel joined us and he introduced a case study. And now and then he gets his friends to deliver interesting ones. We had a fellow from McGill, right?
Yes.
Michael Libman.
Yeah, from up there in Canada.
An interesting case. And now I understand you might have an interesting case as well, Paul.
I do. Thank you very much for having me.
What do you got?
So I have a unusual presentation in a 4-year-old male from New York City, no travel history, the Bronx actually, never left the Bronx, lives in an extended family. There's no history of medication or surgery and no known allergies. And he presents just depressed and lethargic with a little bit of an increased respiratory rate.
Okay.
4 years, that's our youngest patient, isn't it?
That actually is, yeah. we usually deal with it all. So this is a 4-year-old. So a lot of times with 4-year-olds, we get history, say like their caregivers, because the 4-year-old isn't usually going to tell us much. So from the is this 4-year-old, do they have diarrhea, anything like that?
No, he's got an elevated respiratory rate, and on radiographs, it looks like it's pulmonary edema. And none of the extended family is ill. It is the springtime, and on the lab work, he has a elevated white count, which is due to relative lymphocytosis, normal red count, and normal hematocrit, and slightly elevated liver enzymes.
How about eosinophils?
Eosinophils are normal.
Normal.
Okay. So we're not seeing increased eosinophils. So that's helpful. That's given our listeners a clue, maybe.
And this is a recent development in the past month?
Relatively acute onset.
Mm-hmm. And
a little not eating as well as normal.
Okay. And so this 4-year-old's been pretty healthy up until this point?
Up until that point, completely healthy.
Okay.
No medical history.
Mom's probably a little worried.
Of course.
No, any weight loss?
No
weight loss.
Not significant if it is. Don't have exact weights from earlier.
Now, a 4-year-old is not yet going to school, right? But you say he's part of an extended family, so there are other members around.
Yeah, it's a big. There is a social component, but not schools.
Everyone else is okay.
Yes, everyone else.
What do you think, Daniel?
So breathing a little fast, kind of depressed, lethargic.
No diarrhea. No vomiting.
Any fever? Do we know if there's any fever going on? Okay.
I have a funny question, which listeners will understand. Any jaundice?
No.
The whites of the eyes being colored yellow?
No.
Okay, so the whites of the eyes are still white. That's always good. Okay, so we know no past medical problems.
I can't ask any of my sex questions because he's only 4 years old.
You could ask, but they would be as inappropriate as always. Yeah. Maybe more so now. So no surgeries in the past.
Correct.
Okay. Not allergic to anything?
No, either medication or environmental allergies recognized.
Okay. No diseases that we run in the family that we know about? No.
Nope.
Okay.
Healthy family.
And this 4-year-old's not taking any medications, I would imagine. Right. Okay. All right. He lives with this extended family. Probably a lot of exposure, I guess, to non-family members as well.
Yes.
Okay. Would you describe this as a clean environment? We're in the Bronx now.
Yeah, a very clean environment and a lot of outdoor exposure as well. So a lot of fresh air. And,
okay. So is there potential exposure to insects?
Well, sure. Being outside a lot. I mean, it is where he lives. There's exposure to the open air, no screens, so potential exposure to insects.
Okay. But you had said no travel. So this individual has born and grew up in the Bronx. Okay. Have the parents traveled at all?
No, they have not either.
Okay.
Any of this extended family?
No, none of the immediate social contacts have traveled.
Mm-hmm.
And this has not been associated with a change in diet of any kind, right? No. Nothing has changed.
Nope.
Yeah.
What about foods? We always worry a lot about like raw consumption of raw foods, any things like that.
Yeah,
do we have any raw foods?
Raw fish consumption, and there is the only medication actually is vitamin supplementation, vitamin B and vitamin E supplementation.
Okay, so
just as a routine,
taking vitamins.
All
and raw fish exposure. Okay, that's probably important. And so we know no elevation in the eosinophils, so that should help people a little bit. the increase in the respiratory rate, and what looks like pulmonary edema.
Do we have a physical exam at all?
Physical exam is normal.
Normal,
The things I mentioned.
And then you had a few bloods as well, yeah.
Now, you weren't able to feel a spleen, were you?
No.
Okay.
But I presume he has one, right?
Yes, there is a spleen in there, but not palpable.
Okay. That rules out malaria, right?
Well, no, not necessarily. And, and I think we, the interesting different presentations, right?
Yeah.
But we're not in the tropics, right? This person hasn't traveled.
We just had a discussion of Daniel's last case, which was a malaria case in an Indian gentleman. And he said, in the tropics, if you have fever and swollen spleen, it's probably malaria until proven otherwise.
Yeah, that makes a lot of sense.
Makes sense, right? Yeah. But we're not in the tropics here.
Yeah, we're not in the tropics.
So we have to remember, this is New York City, and there's no travel history.
Which is really like being in the tropics, I joke sometimes.
In August.
In August, right.
But people do bring things to New York from elsewhere, of course, right?
Yes.
This is a big port of entry, and there have already been Zika virus infections brought in, and so other things can be brought in as well.
Well, West Nile 10 years ago, or 15 years ago, was a very big emergence.
Huge.
Yeah.
And spread throughout the country over the next years. Yeah, very interesting situation there, which we can talk about more next time because people will understand.
Yeah, and so we went through all your, 4-year-olds. He's not a drinker, not drinking the palm wine like our last individual, but a lot of insect exposure.
And, of course, a 4-year-old is not going to school yet, is not doing a lot of the things that adults would be doing.
No recognized risky behaviors.
No recognized risky behaviors. That's
so
right.
we have this pulmonary thing going on. So something's going on in the lungs.
Right.
Breathing a lot.
Remind us what the bloods were again.
Elevated white blood cell count because of a relative lymphocytosis with a normal hematocrit and white blood cell count.
Okay, so not anemic.
Correct.
Okay. This is a tough one.
Very tough. I wonder if... Too bad Dixon's not here.
You know what? I will tell him you said that. He will be pleased.
It would be interesting to... But he would ask questions that... well...
I guess we should ask more about... Should we ask more about the fish? Is it just fish? Are there any kind of... Should I say frogs? Any things like that?
No, just fish.
Just fish. Okay. All right.
But everyone else is eating the same thing as well.
Yes.
Right?
Yeah, the family eats together.
So that's interesting that only this 4-year-old has this problem. I'm okay. Anything else?
Hopefully that's enough. Hopefully some of our listeners will be able to figure this one out.
Well, we have quite a crew of listeners who chime in and guess. We'd write in and we read them next time. So.
Yeah, I've heard from some people say, "Oh, you guys, your listeners get it right all the time. You should just crowdsource all medical diagnosis.
Yeah.
Be crowdsourced." I actually think we have a pretty sophisticated listener population. So,
yeah,
I think if you could crowdsource all medical problems to our listeners, then maybe that is a good idea.
We have a couple of emails. Do you want to sit through these? Are you okay? You don't have to rush off. All right, we're gonna get back together with Paul.
That's fine. I'm happy to.
In a week, right? And and Paul will solve.
Next week we're gonna go to Paul's location.
All right, so
we'll be traveling. We'll try not to be late.
I'll try and release this as soon as possible because you only have a week to get this. However, we do get in late comments, and this first one is from our friend David, who says, "Dear Twipsters, I started out late for this one, and again, I'm preparing for failure. I still have to listen to the latest TWiP, but I know failure is an inherent part of the learning process. One cannot make an omelet without breaking eggs, and anyone who ever learned a second language will confirm it takes many mistakes before some fluency arises. The same goes for the science of life. The case you presented was seemingly simple, but I went back and forth, and I can't come up with a definite and certain diagnosis." Now, this was the case before last of the farmer in India, remember?
No.
You don't remember? Or?
You'll have to remind me. As he goes, maybe it'll come back to me.
All right. The jaundice, bilirubin levels, and enlarged liver indicate that liver and bile are affected. I dived into liver flukes. Fasciola appeared at first glance to be the most reasonable option, being passed by freshwater snails, but it does not seem to present itself in India. The geographical is way off. Otherwise, this would have been my preferred option. Another parasite that kept coming back was clonarchiasis, but then the man should have eaten raw fish, and there's no mention of it. I tried a symptom checker, and liver cancer of the bile duct topped the list, which again are associated with clonarchiasis and Opisthochus viverrini, the latter one presenting in Southeast Asia, not North India. But as the case was considered a fun case, I hope the diagnostic was not so gloomy. My guess is therefore ascariasis, as it is also associated with fever, jaundice, and can present hepatomegaly in some cases. I guess I really miss a point, especially the darkening of the skin was a symptom I did not encounter for any parasite. I eagerly await the moment that TWiP 108 goes live. Thank you so much for stimulating my curiosity continuously. Waiting for rain in hot and humid Nicaragua. P.S. I called your efforts relentless in an attempt to compliment you. I would certainly be happy if I was labeled relentless in my efforts, but I may well miss the finer intricacies of the English language, not being a native speaker.
Oh, now I recall.
This was Kala Azar.
Kala
right?
Azar. So it was the dark illness, and we mentioned that the original terminology came from the Hindustani.
Yeah.
So that was visceral leishmaniasis.
It's visceral leishmaniasis. That's right. We want to get the next one.
P writes, "Hi, I really enjoy listening to your expertise and continually learn from it. Concerning the case study for 107, I am guessing protozoa with an emphasis on E. histolytica based on several symptoms, including the jaundice, since it is known to invade the liver." And
that's the same case.
Oh, is it the same case?
The farmer from India, right?
All right.
So that's wrong.
Caleb writes.
Yeah, this one is right up your alley. There you go.
Greetings, doctors. I was curious about where I could find a 6th edition of the Parasitic Diseases book. I had a 4th edition of the book, but I gave it to my father-in-law, who is a neuropsychiatrist, who through my talking about your show and becoming extremely interested in parasites. He always tells me that if he were able to go back, he would have become an infectious disease doctor instead of a neurologist. I have found copies of the 5th edition on Amazon, and I would love to have an updated version of the book. I love all your shows and tell everyone I know about them whenever I get the chance. Cheers, Caleb Hubbard, PhD student, medical and veterinary entomology.
Would you know anything about that, Daniel?
Yes, the 6th edition of the book. The 6th edition of the book is actually well underway. It's been completely revised twice, and actually Peter Hotez, people may know from being on the other editions, and Chuck Knersch are going through it as well. And we have a deadline, actually. The end of this month, we're going to try to get it out to a professional editor and try to get it published. Now, we're going to try to make history with this edition. We are going to give this next edition away for free. So we're going to give a copy away to every single medical student in the United States for free. We're also going to give a copy away throughout the third world, the tropics. And then the only version that will be for sale is we may make a hard copy version.
And what's the timing on this?
We're hoping to have it out in the next few months. So it'll go to professional editor at the end of this month, beginning of June.
Wonderful. There you go, Caleb. A little more. Wait a little more.
Hot off the press.
If you want the 5th edition, I'm authorized by Dixon to give you a link to that, so let me know. Just send us another email. The next one's from Anthony, who sends us an article published in PLOS. PLOS 1, large-scale range collapse of Hawaiian forest birds under climate change and the need for 21st century conservation options. Hawaiian forest birds serve as an ideal group to explore the extent of climate change impacts on at-risk species. Avian malaria constrains many remaining Hawaiian forest bird species to high elevations where temperatures are too cool for malaria's life cycle and its principal mosquito vector. I guess the point is that the mosquito range is going up the mountains as climate change occurs, and these birds are going to be not able to get away at some point.
Forced tire.
Right? Wow. To lose any more birds in Hawaii would be tragic. The mosquito challenge because of climate change is something that many people will soon see too. Perhaps the study of an avian disease and the insect vector will be as useful now as it was for Sir Ronald Ross. Dixon and I did a paper a long time ago about how the malaria range is changing in parts of Africa. It's going up in elevation. Because,
and not just malaria. A lot of the vector-borne diseases, as we have climate change, it's going up, it's spreading out, it's changing.
You can take the last one, Daniel.
All right. Hello, everyone. Caroline writes, I just wanted to send a note to Daniel thanking him for calling out the importance of a laboratory in TWiP 108. I've worked in a medical laboratory for 17 years, and we are definitely the under-recognized profession. I've had physicians ask me if we, in quotes, actually needed a degree for that. All certified medical technologists or clinical laboratory scientists, unless they are grandfathered, must have a bachelor's degree and have completed a clinical year in a hospital-based training program, which includes chemistry, toxicology, hematology, blood bank, microbiology, and immunology. Most are ASCP certified. Some have master's degrees. MTs work on pathology, microbiology, and chemistry, doctoral staff. Medical technologists are supported by many highly skilled support staff, such as MLTs, lab assistants, and phlebotomists. So thank you, Daniel. I have included a link with a pretty accurate explanation of medical technology profession. TWiP is my favorite, but I love all four TWiCs: Caroline, MT, ASCP. And yeah, thank you. No, I think I made the comment last time that a physician without a lab, I have like a list of quotes next to my desk, and one of them is a physician, or I should say a clinician without a lab is like a feral dog. It's no, I mean, you need some sort of check, so you know, like, am I out there? Is this really, is this really correct? So you need this for the diagnoses, and you need skilled people working in those labs so you can trust the results. So thanks, Caroline.
That will do it for TWiP 109. You can find it at iTunes and microbe.tv/TWiP. You can also find it on Google Play Music if you have any questions or comments. And we want you to also send in your case guests for this week's case. You can send them to TWiP at microbe.tv. Our mysterious guest for today has been Paul. Thank you very much for coming, Paul. We appreciate it.
Thank you for having me.
We will tell you more about Paul next week when he reveals the case. But in the meantime, please guess. It should be interesting. 4-year-old, our youngest case so far. And next week we can talk all about what Paul does. Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Oh, it's been great. And, but I'm realizing, didn't we have a younger case? Remember that little girl from Australia? How old was she? The little girl that had Ballontidium coli.
Did we do that?
We did a case of a little girl from Australia. I don't think I mentioned she was from Australia. I kept that a secret at the time. But it was a little girl with, remember there was the pig exposure? Her family was out hunting swine.
I don't think she was that young, was she? That was TWiP, either 102 or 106.
101.
Check.
Ballontidium coli, TWiP 101.
Young girl, less than 10 years old. That's what you told us.
So now we're really getting down.
Yeah, that's right. Fever and diarrhea for 2 weeks. That was a long time ago. Well, not really. That was February. For some reason. It seems like a long time ago. I don't even remember. Oh, anyway. Thank you, Daniel.
Oh, you're welcome. Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.